This meeting is being recorded.
Hi, my name is Afshin Beheshti, and I'm currently an investigator at KBR and NASA Ames Research Center, also visiting researcher abroad and president of a nonprofit called COVID-19 International Research Team. So this is going to be a second lecture I'm giving for the We Speak Science series. And this is the first lecture I gave was for you to understand what space biology experiments are done and why even care about space biology and give you an idea of what happens and what type of research is done in space around space biology. So now I want to discuss what kind of resources are available for, let's say, for the data that's out there and freely available for you to actually mine space biology experiments that are previously done. And the key is, it's, as I previously mentioned in the previous lecture, is that it's rather expensive to do these experiments, and not everyone might have, let's say, funded grant money, and students like yourself are just learning and they want to just explore. So there's actually resources out there where you can mine data that is publicly available on your favorite, hypothesis or questions that you develop. So this is done by, doing omics, and I'll get into this in a second. So omics right now, you can actually do this in space, and I'll get into what omics is if you don't know, but there's things you could do like for sequencing, DNA sequencing or RNA sequencing. But, you could actually do that on the International Space Station. There's experiments you could do, once they, let's say, run those experiments that I previously talked about in the last lecture, you could actually now take samples and do it right on the space station that's out, and then get data right away. The other part is, you send samples back to Earth, and then you do all the processing in labs on Earth. So what is omics? Just in case you don't know, omics is basically the idea of you look at the DNA, proteins, or RNA, and then you do sequencing. So you probably heard of DNA sequencing. You take the DNA, you sequence it, and then you find the global profile of how changes happen, versus your experiment versus your control. RNA is, again, you take the RNA, this is an example from a cancer paper that's out here, but I like to show this, like, where you could look at, for example, DNA mutations that might occur. You look at this global sequencing effect of the overall changes might happen. RNA, again, you look at the overall RNA, which is transcriptomes, that could have changed, and then you can look at all different types of RNA if you sequence the global profile. Proteins, the same thing. You do proteomics, it's called. So you take the proteins and you do this massive proteomics, and you look at the global, again, metabolism and so on. So omics is basically, looking at the big data, looking at the big picture, the global profile for all these different factors in biology, and that occurs. So that's what basically omics is. And in omics, you have all these different omics, and then I'll relate, if you do a multi-omics kind of study, you could relate how the overall biology happens, due to some effect like space flight. And then there's, of course, a lot of computational power you have to do, and there's still kind of issues that you have to figure out with omics when you're doing the analysis. So again, what is omics? When you do single omics, I'm sure people have heard of the example of, let's say, there's a few blind people, and they're touching an elephant, and if they don't see the picture, they might, every person, depending on where they're touching, they're going to tell you it's a different thing, but they don't see the whole picture. So when you do this, again, multi-omic approach, look at the DNA, RNA, protein, the global profiling, then you can actually see what's happening. It's a whole elephant. And of course, there's many options for sequencing you can do nowadays, and which I won't get into here. And then you could get the overall idea, depending on cost and resources. You would choose which one. So you might have heard of systems biology. So systems biology is now which is in the past couple of decades, the terminology came out where, and for my definition, this is NIH's definition, the National Institute of Health in the U.S., how they define it, but you could read this here. But my definition is basically, it's like a top-down global approach where not only, do you just look at the biology or deal with biologists, but you use computational approaches, use technology and everything combined together, and use disciplines together. For example, physicists are involved, mathematicians, biologists, chemists, to really get the global feel of what's happening, as opposed to just, having one discipline, one idea happening. So this is where omics come in, and is perfect to look at this larger picture, as systems biology wants to look at, and then you can figure out some key factors happening, and then you can go back to the, as I say here, reductionist biology, looking at the single factors after you've seen the global view of what's happening. So using those approaches is one thing. Is now what the resources you can use, and the main resource I'm going to go over that you guys can actually mine now, as students can mine, is GeneLab, and it's research by NASA called GeneLab. The links here, genelab.nasa.gov, which is a publicly free, available. Anyone can go on here, And this is where all the omics data ends up that people have done from, principal investigators that have grants that run experiments, or people at NASA run experiments, or people at, let's say, in the European Space Station, and that JAXA, some with Japanese Space Agency. Any omics that becomes available and we have permission to put on here, we put on here as available. So that people actually could now mine this data. I'm going to go over in a second what GeneLab is and how to look at it. And you can mine this data with any of your favorite questions that you might have. So the ecosystem of GeneLab is set up so you have all the different organisms that are experiments that are done that I discussed in the previous lecture. For example, from mice to fish to Drosophila or fruit flies, C. elegans, worms, and then you can ask your favorite question, looking at the different omics. For example, what happens with lack of oxygen or oxidative stress, or look at different common tissues that might be, let's say, from the mouse experiments that might be related. Again, you can look at fruit flies and how, for example, some different sequencing are related back to other organisms. And then there's, of course, microbes and plants, and then figure out how these are being impacted together. And then you use that multi-omics approach, look at all the different type of omics that are available on GeneLab, and then find the human relevance. On GeneLab right now, we don't really have much human data other than cell culture that's been flown to space. So because mainly that's due to, when you have sequence data on humans, you would be having an identifiable region or identifiable way to actually know who that person was, depending on the sequence. So usually, for clinical studies that you might have heard of, that people make those data available, you de-identify that. So then now you can't trace that data back to any of the individuals who were part of that study, which is to protect their identity and protect their privacy. As you can imagine, for astronaut data, there's only a few astronauts that are at one time in a mission, 8 astronauts at a time. And for that mission, is the sequencing data, for example, DNA sequencing, RNA sequencing, you can easily trace back to which astronaut is which easily. So the human data is still, pretty private, and you could request it, but it's not on GeneLab. So the GeneLab data is basically set up like this, where, PI principal investigators or other investigators, there's a submission portal where you submit your data. And then what happens is, the raw data, the sequencing data, any types, or the omics data, any of those type of omics data is now put in GeneLab, there's a portal, and then there's, it's made available. And then what happens is a user data workspace that any of you can sign up for, where you can actually work on the GeneLab workspace or download the actual data yourself on your computer and analyze it yourself. So there's a data portal, and then there's like a whole, and then what GeneLab has done and is doing right now is providing resources where you have the raw data, then there's some process data. Eventually, all the data will be processed. So then, if you don't want to or don't know how to analyze our data, you can have the higher order process data, and then the metadata I'll get to in a second. And then there's on GeneLab we have this whole like pipeline analysis tool shed where people can go and use these tools and interactive tutorials to actually analyze stuff on GeneLab itself if you don't have much programming skills. And then there's also a visualization portal where people can actually just go in and type in their key favorite genes, for example, or proteins, and say what's happening based on the data. And that's available to make it really simple to come up with quick and new knowledge. So the whole idea is this, we have the raw data. This is for the data scientists, really hardcore bioinformaticians and programmers, where they just want to download the raw data and analyze it themselves. So the standard biologist, might not have those programming skills. So that we have that kind of data where, you have the higher order data. It's not the raw data. You have, for example, this differential expression data or higher order process data. And then you have you have also pathway changes you can look at. And then we call the citizen scientists or, young scientists for like yourself, students who might not have the skills yet, but we have the data at that stage to actually people can mine, and still you can come up with new knowledge and play around the data, even though you might not have the skills yet to process data as, say, a data scientist or bioinformatician or the biologist would. So there's many different forms of ways you can mine the GeneLab and use it to create some really neat new knowledge. And just because you might not have the experience or skills doesn't mean you can't mine the data and come up with interesting ideas. One thing we have here is that we call it GeneLab power users, which we have these GeneLab analysis working groups, which is comprised of scientists from all around the world volunteer to join these things. And there's four analysis working groups out there. One's the animal, one's multi-omic system biology, which I lead, another's plants, and another's microbes. And we have approximately 250 members spread out through all these groups. But the whole idea is that scientists from around the world, students are welcome to join these, that come together, and then they can actually together come up with new ideas, work together, and come up with some really interesting science, which people have in the past. And in the next lecture, I'll show you some science that has been produced by these groups. But again, students are welcome. All forms of people in different career levels are welcome, from faculty to students and so on. So the type of data we have on GeneLab now is different types of data. So we have more than 350 data sets, almost 400 data sets now on there. A lot of different species that are there. And then this is the amount of data we have on GeneLab if you're interested. But there's actually space flight experiments. Half of them are about that. The rest are like ground simulated experiments. And these are different type of organisms. As I said, the human data is not really astronaut data, but it's cells. So cell culture has been flown to space, or cell culture experiments have been done on Earth. That's mainly that. And then there's microbe data, and then a lot of rodent data, of course, and there's other organisms too. And this is the type of omics that we have that's on it. A lot of it's mainly transcriptomics, the RNA sequencing. And there's different types of RNA sequencing, which I won't get into, but there's the older data, which were microarrays, and now the newer, and then this is a few years ago, a decade or more ago, and then now the technology evolved. You got sequencing data, so now this is it's lower amounts, but this is quickly becoming more and more prominent. So then there's also environmental data. So now you might want to have the questions, like you download your omics data, you want to analyze that, answer your favorite question, but then you also want to know what data sets might be impacted, for example, by space radiation impact. And you want to know the dose they got for that experiment on the ISS. So that data is available here, on GeneLab, if you go environmental data here, where now you can actually look at, let's say this is the data set you're looking at. You go here and you can figure out the dose, let's say, that I got, and then relate it back to the analysis you've done. And then the data visualization. So this is now for, you don't really even have to analyze the data for some of the data sets, that eventually all data sets will have this visualization incorporated when you have the higher data. So you could go there, you can look at these different plots, for example, the PCA's principal component analysis, where you're looking at the clustering of samples, how they might group for this data set. And you can see, for example, these, different groups, how they cluster well together, your controls versus that, and maybe look at spaceflight. You can look at, how well different data sets might group together. And then you can look at, for example, heat maps. So these heat maps is overall comparisons where, this is like different conditions for the space flight. And then, this specific data set had some experiments related with cells being infected, microbes being infected. So then, you can look at that. And then you can look at individual genes in response. For example, just making an example here, you have these flight and ground samples, and you can see that these genes are up regardless, which means that, they're not really impacted by space. So let's say if you typed in your favorite gene and you're exploring and you want to find out what's happening, you have a hypothesis. This is maybe a quick way. You go to the visualization data for that one data set, type in gene X, and then if you see, oh, there's actually a change happening across here versus, flight versus ground, then you can be like, okay, this gene might be some more thing to look at and further explore for my hypothesis and question. Or if it's something like this where it doesn't change over any of the conditions, it's the same, then your hypothesis doesn't work. So then you go back to the drawing board and come up with a new question. And then you can look at the individual genes and see the expression and things like that. So this is how you can explore how you can actually start looking at space biology question, answer, have your favorite question that you want to answer by really just going on GeneLab and either download the data if you have the experience to look at processing data, or just go to the visualization tools and enter your favorite gene or favorite question you have on their pathway, and then be like, okay, what's happening? And then you can come up by yourself some neat exploratory ideas as a project, as a student project, where you can start answering things. And in the key is being creative about it and looking at many different aspects on how to actually explore the idea. In the next lecture, I'm going to give you an example of how to do this. So thank you for your time and listening.